So today we have Martina. She could also be a volunteer, but she can't because she's already finishing her PhD. It's in the last, the last time. I don't know, it's like submitting the PhD in the next weeks. She did her PhD. I think she will explain about what in the at the Mares group, the fish ecology people. And well, now she will explain what she has been doing during these years. Thank you, Martina.
Thank you, Marco, and thank you to come to this seminar. And okay, yeah, I'm Martina, and I'm a PhD student at the Mares group, and my thesis is focused on the individual variability on circadian-related behaviors or chronotypes. And today I want to present a chapter of my thesis that involves.
Sorry.
Yeah.
That's how to. I thought it was my job and I left it. Sorry.
So I want to present a chapter of my thesis that involves the study of chronotypes and personality and the interaction between these behaviors. So we are different. Each of us are different and reacts to the same situation in a different way. And we know that these differences between us are with us personality, no? But what happens with the animals? Animals also show personality, behaves in a different way from the other animals from the population. So the answer is yes. Animals show personality. Here we have two examples of two different squirrels in the same situation and abort the objective to get the nut in different ways. The first one get the nut and go out, and the other one open and eat the nut there. So both of them achieve their objective, and probably if we expose them to the same situation, we act or behave in the same way, and we can consider their behavior as that their behavior is repeatable. So the repeatability is an index that inform or calculate the degree of the consistency in behavioral differences between individuals. So here in the first example, each individual behaves in the same way in each observation. So the differences between them are consistent all the time. Contrary, if each individual behaves in a different way, there are no differences between them, and there are no repeatability. So the index of repeatability is used to detect personality or behavioral types, and personality in animals is studied along 5 different traits: the exploration, that involves the individual willingness to investigate new situations; the activity, that is the frequency of movement; the boldness, that is the individual reaction to a risk situation; the aggressiveness is the agonistic response to specifics; and the sociability is the individual social interactions. And we need to imagine these different traits as a continuum. For example, in the case of boldness, we have in one extreme the bolder individuals, and in the other extreme we have the shyer individuals. And the same for other traits like exploration. And often these behaviors are correlated between them, generating behavioral syndromes. A behavioral syndrome is a correlation between 2 or more behavioral types, where here we have the more exploratory and bolder individuals. And these behaviors can generate between personality and other traits, for example, the home range or the dominance. And in humans, often personality are correlated with chronotypes due to the implications with the human health. So chronotypes are behaviors related with the circadian rhythm, like the waking time defined at the first moment of daily activity, or the rest onset defined at the last moment of daily activity. In other words, is the preference in daily activity time. So for example, the pink individual awakes before the sunset, have their peak of activity in the morning, and then with the sunset they go to bed. Contrary, the green individual awakes later in the morning, have their peak of activity in the afternoon, and go to the bed later. So this behavior is often studied in humans to the correlation with health. But in animals, and are often correlated with personality of humans. But in animals, the problem is that the personality, we need to study personality under controlled conditions, because when we study personality, we want to know the internal expression of the behavior without correlations with environment or social context. But contrary, chronotypes emerge from the internal circadian rhythm in combined with the environment. So we need to study them in natural conditions. And we have some evidence that animals express chronotypes, but the major part of these studies are on terrestrial animals. And what happened with fish? We have a lot of studies or evidence that with the personality of fishes under laboratory conditions, but we don't have any evidence of that fish show chronotypes. Why? Because monitor these individuals in the natural environment is more difficult. But now with the development of acoustic telemetry, we have the tools that we can monitor these individuals in the environment. So the acoustic telemetry is a technology composed by two parts. The first one is an acoustic tag that is implemented in the fish and emits a codified signal, and the acoustic receiver that is placed in the study area. And when a fish enters in the detection range of the acoustic receiver, this recorded their presence. So now we have the tools to can study the personality and chronotype symptoms in whale fish. So the objective of our study was to evaluate the existence of chronotype personality syndromes in a wild marine fish. Firstly, quantify the personality under laboratory conditions, and then monitoring the behavior under natural conditions and can calculate the chronotypes. And we selected the pearly razorfish for different reasons. They live in sandy bottoms of warm waters. They also have an important social implications here in Mallorca, because it's one of the most important target fish for recreative fishers. They show a sexual dimorphism. They born out female and they become male. So females are smaller than males, but also show differences in their coloration, in the shape of their head. And the females presented this clearly pearly spot on their abdomen that allow us to sex them. And also show this particular behavior, this burrowing behavior, that for us is really important, because during at night they burrow in the sun to avoid predators and rest. So through acoustic telemetry, we can obtain a chain of detections like this, where during the day we have a lot of detections, but during the night we don't have detections. So through a hidden Markov model, we can separate the active and rest state, and from this and convert the detection chain in a chain of states. And from this chain of states, we can obtain a waiting time and response to define the chronotypes of the species. But for this study, we focused on the waiting time, because we detect that the response is more related with the sunset and the risk of predation, and what a waiting time defines better the chronotypes of the species. So we went to the mud, we went to the sea to fishing them, because we only can obtain these species fishing them. So we go to fishing them, we transport it to the linear, to the laboratory, and maintain the individuals in individual aquariums for a week. And during this week, we perform 4 personality tests in each fish during 4 consecutive days. For exploration, we performed a novel object test that we introduced a little toy in the aquarium, and we quantified the interaction with the toy, the time out of the sun, time near the toy, and the minimum distance to the toy. And then from these metrics, we combine it to obtain a unique exploration score. For activity, we performed an open file test that is the recorded data activity without human interactions, and we developed the deep learning algorithm to can track the fish into the aquarium, and from this tracking we calculated the covered area, distance traveled, and time out, and combined these metrics to can obtain a unique activity score. For boldness, we simulation a predator attack, and we calculated the latency to exit to the center. And finally, for aggressiveness, we performed the mirror test. You can see that here they are mirror, and we count the number of times that the fish bited the mirror as an aggressiveness score. So at the end of this experimental week, we tag them with acoustic telemetry and release them in the natural environment. And in the sea, we tracked them with acoustic telemetry and recorded their behavior. Then we analyzed the data and we calculated the repeatability of these traits: exploration, activity, boldness, aggressiveness, and awaken time. And then we search for correlations between these traits. So for the results of repeatability regarding the personality axis, here we have the exploration results. Each color represents an individual. The black dot represents the average. And there are, here we have, here we have the less exploratory ones, and here the more exploratory ones. And the repeatability obtained here are the lower obtained in this study, the 0.16, but is significant. So we have behavioral types for exploration. For activity, the same, the less exploratory, the less active individuals and the more active individuals, with a repeatability of 0.24. For the boldness, we obtain the highest repeatability of 0.67, and the shyest individual, this one, lasted 7 hours burrowed in the sun, every test, then the simulation. And finally, for aggressiveness, we obtain a repeatability of 0.53, and this one bites the mirror 300 times per test. For the chronotypes, we obtain a repeatability of 0.62, and we obtain early types individuals and later types individuals, and then we search for correlation between the types. And we found a significant correlation between exploration, boldness, and activity. Here we present that the more active individuals are also the bolder ones. Here there are that the more active individuals are the more exploratory ones, and the more exploratory individuals are also the bolder. So this means that there are syndrome between exploration, activity, and boldness, where the more exploratory individuals are also the more active ones and the bolder ones. But we can find any significant correlation with aggressiveness. But when we search for correlations between personality and chronotypes, we found a significant correlation between aggressiveness and awakening time, where the later type individuals are also the more aggressive ones. But we don't find any significant correlation between chronotype and exploration, activity, or boldness. And in conclusion, we can see that the pearly razorfish exhibit different personalities, and we found or we identified the syndrome between exploration, activity, and boldness. We also provide the first evidence of a correlation between chronotypes and personality in marine fish. Thank you to the combination of our observations under laboratory conditions and in natural conditions. And also, our research shows that the razorfish are the ideal subject to study personality under laboratory conditions and chronotypes in natural environments to can avoid the study of the individual variation. So thank you to come today, and thank you to the RAW team to help me in the experiments of my thesis.
Thank you, Martina. Any questions?
Do you have like a hypothesis or an explanation on why the ones that wake up later are more aggressive? I mean,
Yeah, we have hypotheses. We not conclude anything, but this is the same that it is in humans. Humans with every type are always often more aggressive, and we suppose that, for example, in this species for marenes with a male, with a dominant male and several females, and they don't share the territory with the neighbors, or only females and males share the territory. So we can hypothesize that if you wake up later, you need to be more aggressive to maintain your territory and to acquire food and resources. This is our hypothesis, but we don't conclude.
Yeah, so I'm sure you've had this conversation before, but you know, your method is biased, no? Because you're sampling with your fishing, so you're not sampling the whole spectrum of personalities. So you're missing the shyer one. No? You don't think that's a problem?
No, because it's true that the fishing affect to the select for a specific behavioral type of fishes, but we don't share to see all the spectrum of the population. We only search differences between individuals.
So research question.
So we found differences between individuals, so we can extrapolate that in the population there are more differences than that we are that the ones that we show.
Right. Then so you said there's the chronotype is correlated with, which one was it?
Aggressiveness.
Right, but aggressiveness, activity, exploration, are they not all correlated?
No.
No, how are they not?
No. Because
How is it not correlated? If you're exploring more, you're more active.
exploration and activity is correlated, is here, but exploration and activity is not correlated with aggressiveness.
Right. Okay. And the last thing, I just think it would be really interesting to look at stress coping styles. You know what those are? Because I think that, okay, so they burrow for 6 hours, and the stress response in this fish would be. So basically, what you're looking at is the magnitude of the cortisol response, and how long it takes for that to go back down. So I think it will be really high for those that get really stressed and therefore they hide. And that has ecological implications.
Yeah, sure. But we assume that the stress provocated for the captivity is equal for all species.
I don't think so.
No, but we don't. In this experiment, we can or we don't measure the
I think that
stress.
would be the next step then.
Yeah.
And you have a mass spec here. No, you have a mass spec in the IMEDEA, I think. Mass spectrometer. No? Okay.
But yeah, and I think that the response to stress is also related with their personality.
Oh yeah.
So
Yeah, but you've looked at so many different aspects of personality, and I think this one's missing.
yeah, sure. Yeah, maybe it's the next step
Mhm.
to can conclude that. But this is the first step that never before everyone tested the personality on pearly razorfish.
Yeah, no, you put it forward as a model
Yeah.
species for chronotype and personality, and
Yeah.
you've even used phenotype it and everything. So we know a lot of work on that.
Does sediment, the type, so they bury. Does the type of sediment and their ability to bury into it potentially influence their personalities?
Yeah, we have a lot of proof with different. We prove with different methods, and before this experiment, we found the optimal sediment for the species, because they need to burrow entry and
I have a few questions actually. So one is as methodological. So for these personality traits, you have categories, right? So you have all these dots there, they are classes, right? Going 1, 2, 3, 4, 5. So it's like awakening time would be 7 hour, 8 hour, 9 hour, so on and so forth. So for the awakening time, I understand where it comes from, but for the other ones, how do you determine the bins? I mean, what is the measure that tells you, what is the metric if you wanted to use in the different axes there?
Well, for boldness are the time, the latency time to go out to the shelter. There are seconds. These spend more seconds burrowed than these ones. No, for aggressiveness is the number of bites.
Yes, and for the activity?
For the exploration activity, we perform with the all metrics where we perform a PCA, PCA to obtain a unique score. And this score is explained by the different metrics. Here, for example, this individual spent more time burrowed during the exploration test, have more distance to the toy, and don't interact with the toy. And this, for example, this bite more the toy, spend more time near to the toy.
Okay, so
and the same for the activity.
So wouldn't that then potentially already embed correlations between other traits and these putative traits, because you're using the same type of observation, like biting, for example, or burrowing, or that you use to score other bits. So, for example, if I tend to bite more in the aggressiveness test, then maybe when I explore an object, I will also bite it more. Yeah, but we can. We don't find any significant correlation between exploration and aggressiveness. Well, I have to say this was part of my second question. That
sorry.
I mean, I'm not in your field, as you know, but it seems a bit scattered, the results, right? I mean, I can see some sort of maybe some trends if you squint, but yeah, maybe the thing I see most is, for example, there and there that there are different, there are well separated clouds. So I don't know whether you see the same or you guys have discussed.
No, because we don't found a correlation with plots. We found the correlation with statistical tests, and we found significant correlation between them. We have, I don't have here, but we have the number with the correlation.
When you're defending your thesis, will you be able to argue that your scoring method was the best one available for determining what exploration is? Will you be able to do that? I'm not asking now, but
yeah,
I think it would be a good idea. Yeah. Okay. The last question that I had is more as out of these experiments, and I was wondering. So in your case, you're looking at a fish as a vertebrate, right? It's kind of a complex organism. and you have personalities, is absolutely, I'm absolutely convinced that that's the case. If you start to go, in a sense, to simpler and simpler organisms, do you think that there is a threshold below which you don't see personality emerging anymore?
I don't know. I know that, for example, for some invertebrates, there are personalities in some ant species or in different invertebrate species, there are personality, but I don't know where is the limit for personality. I'm sorry for it.
I have one question is about the clothes before. This is it. The R means monovariability explained for the model. The R is the monovariability explained for the model.
No,
Ah.
it's the differences, is the degree of differences between them. It's an index that from to 0 from 1, where 1 explains the variability, between the differences between individuals. So here, for example, we have an R of 1 because the differences of individuals are consistent across time, and here we have an R for 0 because we don't have differences between them. Okay.
Yeah, I was wondering if you looked at difference in size, gender.
Yeah, we because we can calculate the raw repeatability. It's the repeatability calculated in we did the data, but we can calculated that the adjusted repeatability. The adjusted repeatability is considered some factors, and in this case we consider the day of the experimentation, the size of the fish, and the condition of the fish as an internal index of the.
And do you find any correlation with the personality?
We found a correlation between the day of the experimentation and the exploration test. So in the last days, all individuals are more explorative than in the first days of the. But with the size, we don't find any.
What about gender? Is there any personality differences?
Well, in the species size, it's correlated with sex, because females are smaller than males, and we don't find any significant correlation between males and females.
Are more or less the same age, or I don't know if it's age, or like if they have experience in the sea, like the experience they have as a fish, like I don't know how long they live and these kind of fishes, but like it depends also on the experience that fish have in the ocean, right? Like it has been older and more experience with the aggressive.
The size as in fishes is also related with age. Yeah. So no, we don't have any significant correlation. And to kind of find the age of the fish, we need to kill them. Yeah. But yeah, it's related with the size.
So they could have learned, like some animals, maybe they learn during their life.
Yeah.
Yeah, one thing is that when you show the correlation plots in exploration, there is a large difference between, like, you can see like the cluster of points, but when you show the plot of like the continuum
here,
yeah, but when you show the two before the slide, before slide, the slide before, yeah, that one. You don't, here I don't see that, I see like more a continuous spread, so I'm not understanding where do you get like that difference in exploration. Because I mean, the
I don't
points
understand.
that you are showing after,
See?
those are the points from here, right?
No, here,
No, on the
Here?
Yeah, here and then in the
we plotted the mean of each individual
yeah.
for the exploration and activity to searching correlation between
And that
them.
mean value is not the mean value that you get you are showing.
Yeah. But here there are, but here there are not only the exploration, there are also the activity means.
Yeah. No, but I mean, but in exploration, you have like a gap. You have a lot of
Okay,
low
yeah,
values of exploration and a lot of these that have like medium high values.
Yeah.
And in
And
that
here,
in that chart, I'm not seeing that
because it's more a continuum. I think it's because it's the correlation, because when you plot the correlation is
No, con los puntos vitales tendrían que falta de algo.
sí,
Sí, que societat que falta.
sí, no me da
No sembla que sigui el mateix. Però,
but they are the same.
però són els punts de cada individu. No, és la
mitja.
mitja. No són, és veritat que no semblen els mateixos.
Però sí ho són.
Okay, and in case that one is this one is okay. Have you considered like because here you can see like there are like low points from the considering you look at the difference between what are the difference between the fishes that are in this side and the fishes that are in this side because it's like there is like a clear difference between them. Like analyzing these two data sets separately.
Sí. Yeah.
If you have considered it.
No, we no, because we consider each trait as a continuum. So yeah, we found differences between these two classes, but not between these classes. Yeah.
Can they be the points of every test? Because there are a lot of points. There are more points than individuals.
Yeah. It should be 50 points.
Yeah, I should check.
Yes.
Maybe it's per test.
Yeah, it's per test. It's not the mean. It's not the mean. It's not the average. Is the
So it's the actual data.
yeah.
Per test.
So todas.
On the same graph that Danny said, exploration versus activity, it's a bit confusing the slope you find, because if you take separately the two clusters, the top cluster will have a negative slope, or it looks like, and the other one will have a main slope of 0. So while in the end you have if you just look at the slope, it's clearly the more explorative, the more activity you will find. But if you separate the two clusters, it's not sure you will find a positive.
Yeah, probably, but you tested the personality of the population, and the behavior is a continuum, so we cannot separate the two clusters. But
Yeah,
I'm not sure that.
It's not showing that in this case, right?
Yeah.
Because you have
No,
low
in
influence.
this exploration there is not showing you not continue.
Yeah, I think it's because the exploration is the lower repeatability, is the lower repeatability obtained here, because there are a lot of each individual have a lot of individual variability.
And not only that because they have or either very low or very high.
Yeah.
So it's what you
Yeah.
see that are those individuals
Each-
which
yeah.
are separated.
Here, the form of these plots indicate the variability of the individual. So these individuals, the lower exploratory individuals, present a high variability.
The correlations are done with the average for individuals. Right?
No, with the all data.
I think that the aim is to understand the personality of each individual.
Yeah, but you need to, the correlation, you need to be calculated with the all data.
Yeah, the data is not independent. Points are not independent. If they belong to the same individual.
Yeah, but the individual is included in the model So we include the individual in the model to can view the effect of the individual.
This kind of
And
multiple models.
Yeah.
It's a random effect, I would say.
yeah, a random effect. Yeah. And this is a bit more complicated because you need to find this correlation in each individual. So here I show the correlation in the population, but to find significant behavioral syndrome, you need to find the correlation in each individual. So these individuals, the activity and exploration of these individuals are correlated, and in all individuals are correlated these two traits. So in the population, these two traits are correlated.
But in this case, I think that it will be better to use the average of each individual. Because if not, you can have one point of one individual here and the same point somewhere else. It's strange, no?
Probably, yeah.
And also as a random effect, it will be strange, and like the model will expect that there is.
But I think the problem is the plot, no, is the model. Yeah. Because
Yeah, also something is lacking here. There's also a bit of the fit of the model, like the R square, the p-value, also the effect, the size of the effect. Because with the points, it's difficult to defend this a bit in some of the plots, no? Because I feel that there is
But the plots are not an output of the model. Yeah.
No. Yeah.
but the effect is the one you extract from the model. The slope is the output of the model.
No,
So what is the line?
it's a random regression.
There's I mean, the line is a model.
No,
So what is the model? What is the line?
it's a random because you don't extract the
No, at the purple line.
yeah, but it's not the model that we. It's a simpler random regression, and we don't found the correlations with the random regression.
You have a number
For this correlation, yes.
and still plot it and.
Yeah, probably is a better way. Plot it.
Now seeing these plots, I feel that there might be other approaches that you could use, like clustering somehow individuals and then checking if their scores differ. I don't know, because it seems that there are like clouds of individuals that differ clearly.
Probably we can. Yeah.
Maybe for future works when they ask you the question.
Yeah.
Methodology questions.
Mhm.
You only did this for one season, or did you do it for like a whole year?
No, we made it during the spring and the early summer. Yeah, because during winter, fishing this species is very difficult because they are spent the major part of the day burrowing in the sun. So yeah, we target them during the sprinter and summer. Yeah.
Follow-up question to that one. Do you think you would see a difference in their personality if you sample different seasons?
I think that in the personality, no. But maybe the chronotype, well, in other work, we see that the chronotype are very influenced for the reproduction season. So maybe, the chronotype under natural conditions maybe change during the seasons, but personality, no, because it's in the lab. So it's for that we studied in the lab, because we removed the environmental and social interaction.
Question two, is there a mortality rate assumed with the fish that you captured, not all the fish I assume are going to survive through the aquarium?
From the aquaria survive the simple, the 100%.
Okay.
And from the fishing, yeah, we have a mortality rate, but I don't know exactly which is. Yeah.
You mentioned earlier that when you noticed that their behavior was kind of equalizing, you kept them for Saturdays?
We catch them on Fridays, and then we give them 2 days to acclimatization to the laboratory, and then on Monday we start the personality test until Thursday, and on the next Friday we tag them and release them.
Why did you, if you noticed they were kind of decreasing even from that Monday to Thursday, you mentioned Monday they were more aggressive, by Thursday they seem to be. So Monday, Thursday, did you try to sustain any a little bit longer to see if you still see that?
Yeah, the problem here was to that we need to test the individuals. We can't test all individuals at the same time because the space in the laboratory are limited. So we need to perform different tests or different fishings, and we have a very limited time. So probably if we tested with a long period of acclimatization, we have different results, but we search for differences between individuals. So probably in different time or with more acclimatization, we found different results but there are differences between individuals.
Last question. With the toy, you said you put in a toy and you saw their interaction with it. Did you use the same toy all the time? Okay.
No, we have we have different toys, and we presented a new toy every day to ensure that is a new object every day.
Great, so should we stop here and have some beers in Agora? So thank you very much.
Thank you.